Professor Tymish Y.Ohulchanskyy from Shenzhen University, College of Physics and Optoelectronics Engineering. Unfortunately, we have some delay. In this moment, we should be finish this first talk. So my suggestion is that we will try to compensate this delay by shortening, unfortunately, each talks for about 5 minutes, maybe. So we will be in time at the end of our session. So do not waste time, please, Professor Y.Ohulchanskyy. We will.
Okay. Yeah. Can you hear me?
Yes.
Yeah. Okay. Thank you for introduction, and I would like to thank also to organizing committee for providing me with opportunity to talk to present this talk at the conference. And unfortunately, I was not able to arrive to Lviv. So the topic of my today's talk is infrared emitting nanoparticles as agents for biomedical imaging, optical sensing, and therapy. And I represent College of Physics and Optoelectronic Engineering of Shenzhen University, Shenzhen, China. So this is, you can see, Shenzhen University, and this is campus of Shenzhen University in city of Shenzhen. This is new, relatively new, as you can see, university. It was organized only in 1983 as also as city of Shenzhen. And city of Shenzhen is in the south of China. It's in Guangdong province, and it is next to the Hong Kong special area. Hong Kong is here and Shenzhen is here. So this is pretty nice and modern city, as you can see here. And this is once again campus of our university, one of 3 campuses actually. This is North Campus and this is South Campus. And this is actually building of our college here, you can see. And this is, by the way, this is main headquarters of Tencent Corporation. If you can see, there is software WeChat, and also which actually have more than 1 billion users all over the world. And you can see that Hong Kong is like there. Okay, so here is the outline of my topic, and I will start from background and motivation for this work, what I will be presented today. So around one decade ago, a term theranostic was coined, which means combination of diagnostics and therapy. And for therapeutic and diagnostic purpose, for clinics, for healthcare. And nanoscience provide powerful platform which can combine in this nano platform therapeutic and diagnostic features on the same platform. And I am working everything because I have background in optics, and I graduated from physics department of Kyiv Taras Shevchenko University. So in my work, I'm dealing with optical imaging and light enacting therapy, and this is using different nano platforms for this purpose. And if we are talking about theranostics, so this is example of the really old and simplest phototheranostic approach, which still is in clinics. That means that people looking at optically trying to detect malignant tissues, like here cancer, with fluorescence, and then treat with photosensitive drug, and then treat with light, and as a result of so-called photodynamic therapy, cancer cells killed. So another background for my work is a recent advantage in study of optical properties of biological tissue, namely extending optical range into near-infrared range. As you can see here, there is characteristics of optical transparency and attenuation. And you can see that there are so-called optical transparency window in the near-infrared range, near yard 1, near 2, near 3. And also people call it as SWIR, which means short-wave infrared, and also manufacturers of cameras, imaging cameras, also call it as short-wave infrared. That's why we called it near-infrared slash or dash short-wave infrared range. We did previously some work about characterization of these optical tissues, and that was published 2 years ago, and we could see really that in this range about 1.5 micron attenuation is the smallest attenuation of light, and penetration is the penetration depth is the largest. So we are interested to move into this range, into near yard 2 window or a near yard 3 window, with the aim to see deeper what's going on into biological tissues. And this is also a near illustration how near-infrared, short-wave infrared imaging helps to see many things. You can see the same person, the same lady, and photo taken in visible near-infrared and short-wave infrared ranges, and you can see difference. And something can be seen, which you cannot see with visible light. And also you can see difference in this apple, and you can see also difference in visible when you have some fog and you cannot see because high scattering. And scattering is much less for near and short-wave infrared range, and that's why we can see much better image. And that's idea, that's the same we can see with imaging of biological tissue. And also we are working with short-wave infrared emitting nanoprobes, and to make photoluminescence imaging in this near-infrared and short-wave infrared range possible. And people working, this is also from paper of Chemical Society reviews, dye-based organic nanoparticles, rare earth doped nanoparticles. Rare earth ions has very interesting system of electronic levels, which allows for very distinct and characteristic photoluminescence emission, which can help to get really interesting imaging. And also quantum dots and single volt carbon nanotubes. This is main classes which people define by now. Now I'm starting talking about our work in this area. So the first is about rare earth doped fluoride nanocrystals, a nanoprobe for down conversion and up conversion, photoluminescence, bioimaging, and sensing. So first I will start as an example of a paper published in 2018 that was done with collaboration with Harbin Institute of Technology. They synthesize these nice nanoparticles in collaboration with us. This is my old friend and colleague, and we are working a lot in this area. So, and you can see photoluminescence, that it can see up conversion, so at excitation at 800 nanometers, it shows photoluminescence in down conversion, so Stokes shifted, and up conversion, anti-Stokes shifted. And this is optical characteristics and scheme of electronic levels in these ions, and explaining what's going on in this nanoparticle. So this can be read with more details in this paper. And this is some example of that image which also was published. So this is emission of this is cancer cells, and cancer cells treated with targeting cancer nanoparticles, and that's photoluminescence at 1.5 micron. We can do that microscopy, and we can do also in vivo injection. We demonstrated this. You can see that the injection was here, subcutaneous injection, and we can see photoluminescence in that range. And also demonstration how light can be seen through depths of biological tissue. So it can be seen at a few millimeters depth without problem. Another application, this is about of similar nanoparticles, near emitting neodymium and ytterbium doped rare earth nanoparticles for non-invasive temperature sensing. Here we have also distinguished, this is also published this year, and we have two bands of photoluminescence. One is from neodymium and was from ytterbium. And while in exciting neodymium, energy transfer within nanocrystals exists to ytterbium ions. And this energy transfer is temperature dependent. And that's why its relative intensity of this peak at around 1 micron changes if in relation to that neodymium peak. That's why we can use this for measurements of temperature, local measurements of temperature by this radiometric luminescence measurements. And we demonstrated this with process, this temperature sensing during photopolymerization process. We also demonstrated that we can see this ratio changes during photopolymerization with visible light for 80 nanometers, and the same we exciting with for 800 nanometers, and then we can see this photoluminescence and can detect the temperature changes. And that was done in dental application. We demonstrated here that cement, dental cement doped with nanoparticles, and this is on tooth sample. And also some, this is veneer so-called, which people used for cosmetic purposes in dentistry. So this cement veneer is fixed on this tooth, and that we can use during photopolymerization. The temperature should not go too high, and that we demonstrated here. And we also show that the same emission can be used for imaging. So proof of concept we reported here that these nanoparticles can be at the same time used for near-infrared imaging, photopolymerization, and temperature sensing. So next, I'm trying to be to accelerate my talk as fast as possible. So this is second or third point of my talk is near-infrared shortwave infrared dye-loaded polymer nanoparticles for imaging and imaging-guided photodynamic therapy of cancer. Some time ago, we this is collaboration with people from Dr. Vretik from chemistry department of Kyiv University, Taras Shevchenko University, and Dr. Slaminsky from Institute of Organic Chemistry of NAS of Ukraine. And these nanoparticles, core shell nanoparticles, were synthesized in chemistry department. And we developed way how to load these core shell nanoparticles with organic near-infrared dyes, with different dyes. That was also published. And this, as I can see, this is recent results, which showing this actually in preparation manuscript. You can see how this imaging, here this is in vivo injection. And you can see that this is in vivo leg, a mouse leg, and you can see that photoluminescence arising by these nanoparticles goes throughout all body, and you can see all blood vessels. And this is also showing this whole body imaging, starting from, and you can see that different organs pick up these nanoparticles in time. I forgot to say that this is accelerated imaging. This is 2 times, and that leg was accelerated 4 times. So time scale. Yeah, so as you can see, this is liver, this is spleen, and this is heart. So this is in end of, and you can see still also some blood system, blood vessels. I will probably will not talk about this, and switch to another situation, talking about these nanoparticles, both organic dye nanoparticles and rare earth-dot nanoparticles, can be imaged or multiplexed during whole body imaging, biological imaging, using multispectral imaging, photoluminescence with help of tunable here, liquid crystal tunable filter we were installed in front of camera, and that's allowed us to distinguish two type of nanoparticles which cannot be distinguished in normal imaging because they have emission in the same spectral range, but they have different spectral shape, their luminescence. So we were able to demonstrate this in vivo also, and this published in this paper last year and the year before. And now I will be talking about using nanoparticles for imaging-guided photodynamic therapy. Photodynamic therapy, this is photo-induced therapy of cancer and other diseases, which has as a basis photophysical excitation of special molecule of photosensitizer, and there that you can see Jablonski diagram of photosynthesizer, light absorption, then intersystem crossing to triplet level, and then actually from triplet level can molecular oxygen can be sensitized, and that excited singlet oxygen can damage cancer cells and kill them. And it's important also to see a so-called see, treat, and see approach, when you can first detect the tumor and then apply light to treat it, and then see progress of that tumor treatment. And here we also, in collaboration with chemistry department of Kyiv University, we developed this nanoparticle. They developed this nanoparticle, but we developed this formulation with co-loaded with near-infrared dye at photosensitizer. There is also, this is published in this year, and we loaded near-infrared at shortwave or shortwave infrared dye, characterized it spectrally, treated in cells, cancer cells in vitro, and showed that this depending on what type of dye, this formulation works better and kill cancer cell more efficiently. And then also we did in vivo imaging and showing that these nanoparticles loaded with near-infrared dye and also with photosensitizer remains into tumor for, as you can see, for 60 hours. Still we can track it, while without nanoparticles it's actually removed from body by that time. So that's help to really to treat more efficiently. Last part of my talk today will be about nanoliposomes. And why nanoliposomes? What we also, this is another nano platform what we're using for in vivo imaging and therapy. And this is important because, why nanoliposomes? Because doesn't matter how many different formulations of nanoparticles are being right now being developed in all over the world. But liposomes is still one of the major things, and this is clinicians really consider and like them, because this is very biocompatible and everybody knows about this. And actually that's only clinical liposomal photosensitizer in terms of PDT are available. For example, this is photosensitizer benzoporphyrin derivative. It is with liposomes, it is Visidin, the brand name, which is being used in clinics for treatment of cancer, some types of cancer. And and you can see different types of liposomal drugs available on the market. So here we are developing some formulation for PDT and imaging-guided PDT, and here this some steps in PDT. And this is a feature which efficient PDT drug must have here. And as you can see, there is important single oxygen production ability, which actually related to intersystem crossing in the molecule of photosensitizer. And to improve this intersystem crossing, we developed the following formulation. The following formulation, nanoliposomes, which co-encapsulate CT contrast agent and photosensitizer for enhanced imaging-guided photodynamic therapy. This is, you can see, TM of this formulation, and this is formulation what we developed, and it contained commercial and approved computer tomography contrast agents, and it also contains very well-known photosensitizer. And we found that combining these two things, we bring new feature, because iodine in these molecules of this heavy atom effect produce heavy atom effect on the molecule of photosensitizer, which helps to, and this is also showing this liposomes, and this is our TM. You can see that really structure of these liposomes on our team very similar to that image what we can find online. Okay, so next, this is some data, what I was talking about, that presence of iodine in that molecule affect on the single oxygen generation. This is spectral data, and this is in vitro imaging data, and this is in vitro therapy. And we can see that with our formulation, cancer cell survival is the lowest. And actually, that was also published last year, and we at the same time demonstrated tumor accumulation of this. This is passive uptake of tumor of these nanoliposomes, and can be shown by computer tomography and also by near-infrared imaging. And also we found that this is really, you can see this representative image of this pre-treatment, this row of mice treated with light and our photodynamic formulation. This is pre-treatment, and this is post-treatment, almost 3 weeks after treatment. And if you can see, with our formulation, there is no practically tumor, only scar here, but all control groups has still huge tumor growing. And then you can see this is our formulation. So we believe this is, so the beauty of this formulation that it combines only approved formulation. And the same we did with recently published paper where we combined here, just this year, combined this iodinated contrast agent and also indocyanine green, which is only near-infrared dye fluorescent, which is also allowed for use in human. And when we combined in this biocompatible liposomal formulation, here you see nice TM of this formulation. We can see contrast. This is contrast of computer X-ray contrast here demonstrated. And here we can see efficiency in single dosage generation used by spectroscopically determined. And also in our formulation showed the most efficient treatment of cancer cells in vitro. And instead of conclusion, I can see that what we are working on the different photo nanotheranostics approaches for different diseases, including and not only cancer, and working in this using these optical windows, and also developing different advanced imaging technologies. So I would like to acknowledgement of my group and my collaborator from Shenzhen University, and from collaborators from Harbin Institute of Technology, University of Macau, from Taras Shevchenko National University of Kyiv, from Institute of Organic Chemistry, and also this work contains some materials, some data which were obtained with our Brazil collaboration. I also appreciate funding, my funding from Shenzhen and from National Science Foundation of China. Okay, thank you. Thank you for your attention.
Thank you, Professor Ohulchanskyy, for the very interesting talk on the edge of medicine, biology, and what is important with very nice physics. So we have, unfortunately, we have not time almost for the questions. In the chat of YouTube translation, we have two questions. If you answer very, very shortly,
Briefly.
then
Yep.
First question from Leonid Dolgov, Dr. Leonid Dolgov. Could you please...
I'm sorry, Mr. Professor Yatsenko, there is Oksana for reading question, and there are three questions. Okay.
Okay, let's give the answers.
Okay, thank you very much for a very nice presentation. And there are three questions which comes for us from chat. The first is from Dr. Leonid Dolgov from Estonia. He asked, "Dear Tymish, could you please say how you achieved so nice monodispersity of nanoparticles? Could you tell which are the highest quantum yield you obtain for them? For RB3 plus and NERDI 3 plus."
So, see, it's really a question, as I understand your question is concerning about rare earth doped nanoparticles. Rare earth doped nanoparticles about monodispersity, because definitely question is different nanoparticles have different polydispersity, and really this you know, there is chemistry different. You can read in the paper. This is pretty well developed. There are many research group in China working with rare earth doped nanoparticles, and they all getting very nice, very monodisperse, very beautiful nanocrystals. So this challenge, chemical change, is solved. So this is not a question. In regard to quantum yield, it also depends. So from material, but for example, I don't exactly remember about these particles, but I remember that one formulation what we have about neodymium, we have in organic solvent quantum yield around 40% of down conversion. So in this case, it's much easier to obtain high quantum yield for Stokes down conversion photoluminescence. And because of this this is core shell. People developed different structure, core shell, you know, multiple, they're playing with nanotechnology, and quantum yield is in tens of percent for sure of this emission. Okay.
Okay, so
Thank you very much for this. And there is also some additional part of this question. How long these particles are stable in time?
Also, if the question about rare earth doped, because I was talking about different nanoparticles, if the question about rare earths, they are very stable. They are much more stable than quantum dots. They are, because this is inorganic, this is crystals. So we consider that they, during at least we were able to see, once we were doing experiments with mice and remain them, and we could see that some nanoparticles could be seen 1 month after injection. But after, when we saw already half a year, we did not see, but I think that the nanoparticles with time are just excreted from the body. But stability, shelf stability, if you are talking about photoluminescence stability, this is ever. If it's talk about colloidal stability, it depends on different, but still it's actually months. Colloidal stabilities in terms of months, you can store it in shelf.
Thank you very much. And there is one more question from user under nickname Head Scientist. And he asks that, what about the long-term consequences of infrared radiation of human tissues? Is it possible that infrared radiation with high intensity can damage tissue of cells?
This is really in another, of course, it is not here because this conference is about nano, but we are working also on effects on near-infrared radiation. And of course, this is not some ionizing irradiation, because we are dealing with sun irradiation. There is a lot of part of infrared radiation in sun, so everybody goes outside and under the sun. But of course, and we are not using high power or excitation, in this case for photoluminescence emissions. So through all body, we can have just, this is order of milliwatt per centimeter square, and this is not too high. This is, but of course there are some effects about, if you can see, about some low level light therapy or photobiomodulation. We are working in that area too. So, but this is another story. But there is no kind of strong effect, which immediate effect. This is for sure, because our irradiation power density is comparable of that with external, with sun, for example.
Okay, let's interrupt you. We are out of time, and thank you again for the very interesting talks and the answers for the questions. And we have go to the next talk.
Okay, thank you for your attention once again.